Thank you very much for the introduction and for the organization. So as it was just mentioned, this was part of a summer internship following the last year's review panel. And how do you guide someone through a whole scientific process in a 3-month internship? It's not just that we come up with an idea that we want to test, we design an experiment, we collect the data, we analyze the data, but we also need to communicate the results and write them up and publish them. And so Bargibby did that over the last few months, and so please address all the questions to her. That's really her work today. The other thing is it's not a core concept article, so it's not a review. It's actually an experiment and data that we collected in Bob Knight's lab, and that we also submitted to an adult journal where we got reviews too. And we can chat about that later.
Okay, so I'm going to start off with like a basic history, background of brainwaves. So brainwaves were discovered by a psychiatrist named Hans Berger in 1924. So he did this by placing electrodes on a subject's head and amplifying it and then taking pictures. In the pictures, he observed curves with regular fluctuations. This was the discovery of EEG, or electroencephalogram. Electroencephalogram is the electrical activity in the brain and brainwaves. These are also called oscillations. Brainwaves are generally in the form of sine waves. Up there, the ECG trace and the 10 Hertz sine waves are there for reference. So, there are 3 basic measurements in a wave. The frequency is what shows how many times the wave repeats itself in a certain time range. The amplitude is the deflection from the 0 baseline. The phase describes the momentary position of the wave in its cycle. So waves are typically measured in microvolts. The most prominent wave is the alpha wave. This alpha occurs at 10 times per second, and it's generally 20 to 100 microvolts in size. So previous experiments showed that certain frequencies are related to distinct functions. In this case, for the alpha wave, the visual perception is related to the alpha wave. Along with the frequency, the amplitude and the phase are also influential for visual perception. So today, after Berger's research, there were many more experiments which demonstrated that specific brain waves are related to sensory processing and higher cognitive function. So scientists have hypothesized that brain waves are not just necessarily a correlate of the brain at work, but that they might actually be causing or driving how the brain works. So we wanted to establish a direct link, so we were going to demonstrate the relationship in reverse. So our question was, can we modulate the brainwave so that we can study how perception and behavior change according to that? Our experiment. So we took 20 healthy subjects, and each subject went through 800 trials. They were shown 14 triangles in different orientations, so either up, right, or left, and it was in the center of a black screen. So the sequence always ended with a left, top, right cue for the participant so that they could do a detection task. If you can see the left, top, and right, it's a circular motion, so it'll go clockwise. The target itself was a downward-facing triangle. So this was shown after a variable time delay of anywhere from 34 to 850 milliseconds with 25 equal steps. So the triangles were flickered either at a 10 Hertz rhythm or arrhythmically. On every trial, the subject had to indicate whether they saw the target or not, and the subjective visibility changes depending on the alpha phase in the brain. So the target stimulus is always the same, we didn't change it, but what we perceive, what we see is different. So we hypothesized that depending on where the target happens along the cycle, the detection performance will vary. Our results. So we first analyzed the target detection accuracy across all 25 points. So after the rhythmic sequence, we got an average of 75.1% accuracy. And after the arrhythmic sequence, we got 74.38%. So with the group variations or standard deviations, they were along 11 to 12.5%. So we can see that there's not really a big change between the two groups. So basically the performance was the same whether we showed the 10 hertz wave or not. Then we looked at the target detection performance over each of the 25 time bins. So the accuracy does change across the different points, as well as within the two conditions. You can see that the blue line goes up, down, it goes all over the place. It goes from 69-ish to like 81% maybe, and the red line is also going, the arrhythmic is also moving along differently. So to see the relative variation, we took the difference between the blue rhythmic line and the red arrhythmic line, and we produced a black graph on that opposite side. The graph goes up and down, it fluctuates, as you can see. So we fit in a sine wave, and if you look at the sine wave and you count the number of peaks, you can see that it's pretty close to 10 hertz, which is the alpha oscillation. So our conclusions. These results demonstrated that the visual perception was not equal across time, but it fluctuates. So this leads us to conclude that our perception changes rhythmically over time. In this visual exercise, the fluctuation was approximated to 10 hertz sine wave, or the alpha frequency. So it also suggests that the visual systems in humans samples the environment rhythmically at around the alpha rhythm. Our data also suggests that the rhythmic flicker directly modulated the visual system and the behavior, which establishes a causal relationship like we had mentioned in our hypothesis, saying that brain waves drive the perception. So basically, overall, our brain takes snapshots and puts this together so that we perceive it as a movie almost. It's just like a flip book if you think about it. In a flip book, you have pictures on each page, and when you go through fast enough, if you flip through fast enough, it all puts it together like an animation, and you look at it continuously. So this is what we call a discrete perception. After this, future studies will have to investigate how this happens. How does the brain put everything together? How do we see it as a movie and not just little pictures in a book? Thank you.
Krishna, I think it's your turn.
Okay. I want you to answer it, so.
Okay, for sure.
In the paper, you say that performance is better at the trough of the wave. And how do we know this? And then in the figure you showed earlier, wouldn't it be better when you showed the light gray triangles on the peaks and the dark gray on the valleys?
All right, let's go back to the figure. So first of all, we don't do this experiment just with behavior, but we get the EEG simultaneously. And so when you think of the EEG and not of the behavior, you cycle between states of high and low excitability. And whenever you're in the brain at a low phase, then it's highly excitable. And your performance is actually better. But when the brain is not excitable, your performance goes down. So in this case, here, we show the behavior over time, which goes better and worse. If we would show the same graph for the EEG, then it would be the opposite sine wave. You're right. We could add that.
And the first question that he asked, let's not forget that one. Do you want to ask again? That was oh, why performance is better at the trough. Oh, I see what you mean. Yeah. She didn't answer that, did she? Did she? Okay, all right, well. Sorry, my mind was wandering.
We know to do simultaneous EEG too.
Right.
So your behavior and brain, you try to bring those together.
Great. Skylar, did you have a question? No? Sabine?
Is this why people love and it's easy to learn music that has rhythm?
Well, yes. So it's like this is what you call in training. When the brain hears a rhythm, it starts following along with that. So yes, when you hear a rhythm in music, your brain is going to automatically start clapping. Like it's like a clap. You're gonna entrain yourself into that rhythm. So yes.
So when you think of the brain waves, they're there and they have a function, and one of the functions how we think of them is that they help to cut things into pieces and structure them. And so music is one example. It gives you a temporal reference frame, a temporal wave unit that are regular and they repeat itself. And so it's easier to structure information or your surrounding world in those discrete snapshots.
Any other questions from our panel? Darius?
In other senses, such as hearing or feeling or taste, how would rhythms be applied to those, and have you discovered frequencies for those different senses?
So this is a unitary phenomenon across most of our senses. There's one exception. And one thing why those rhythms actually help, when you think of the cortex being like a muscle that you exercise, and you lift something and you have to release it. You lift it and you have to release it. If you lift it all the time, you might get tired over time, and you might not be able to do that for a sustained period of time. And so the cycling is something that every sensory modality does. So for example, you see the same 10 hertz rhythm when you do the experiment in the auditory domain for hearing. You get the same for somatic sensation when you give like little. When you touch like your fingers, for example, it's also not the same over time. Different rhythms have different roles in the brain. The memory system, that's what we talked about last year, has a prominent rhythm at like 5, 6, or 7 cycles per second. And that helps to that's why our short-term memory, or why we think that our short-term memory is limited to maximum 7 items, because there's a rhythm in the brain that facilitates that.
Hasten or Wyatt, any last comments? No? Okay. Yes or no? Yes.
So when you said that like you flash all of the triangles and what ended up happening was they saw a downward triangle, if you were to just flash part of the pattern, would it come out the same?
So you mean part of the sequence, not the entire sequence, but just some of it?
Mm-hmm.
I mean, the sequence is to train the brain, so it would be the same because you have as long as it's at the same rhythm, as long as it's at 10 hertz, it should be the same, even if it is less triangles.
It's an excellent question. We actually did that in the experiment. So there's a control condition where we don't have the sequence in the end that tells you that the target is coming up. And what happens is that this 10 hertz rhythm is even more pronounced because there's no other prior information that you can use. So the visual system kind of flies blind and just does what it does. It samples at 10 hertz. When you have the sequence, it tells you something about it. So it's actually less pronounced in this case, but we choose it because it's nicer for illustration to have like an additional cue that there might be a target coming up.
So I'm going to ask Krishna to abstain here because of potential conflict of interest. And so for the rest of the panel, are we accepting minor revision, major revision, or rejecting? I got a few thumbs up, accept. Okay. Congratulations. Do you want to tell us about your review process with the other journal?
So we submitted the paper to the other journal and waited for like 2 months to get it back. And we got in written text 15 pages back, and the allowance to resubmit, which in the world of adult journals is a minor success, I guess. But this was so much more pleasant. So much more pleasant.
That's great. Wyatt, do you have one more final word?
I had actually made a mistake on the last one. I gotten your article confused with the mind wandering one. So my review on the mind wandering one, just could you switch your two around?
So as the editorial board, we'll talk about that. We'll work that out with the admin. Yes, but thank you. It's great, thank you.